So welcome to the fourth seminar of the seminar series organized by STAPA. And I just want to say that the STAPA is the organization of young researchers and postdocs, and we organize different events to help integrating people, new people and newcomers, and to the people already here. We organize the classic social hour that was before called the beer hour, and also other scientific events like this series of seminars. We had before Stephen Friend, that is the president of Sage Bio Networks. He now moved to create the biomedical part of Apple. I got those news. We had second Thomas Boehm that just arrived here. And he gave us a very interesting talk about host ambient interaction and how that defines the biological individuality in the context of the immune system. And later we have Antonio Lascano from the UNAM, who told us about, he made a very nice historical review about the RNA world hypothesis in the context of origin of life. And now it's a big pleasure to introduce to you Tomas Boehm, that is sitting there. And he is the director of the Max Planck Institute of Immunobiology and Epigenetics in Freiburg, an institute where I did my PhD. Thomas is a medical doctor. He obtained his he studied medicine in the University of Frankfurt, right? That's right. And he then worked as a research assistant in pediatrics in the University Hospital of Frankfurt. Later, he was a researcher and then a guest researcher and then research assistant in the University of Cambridge. And later, he moved to Freiburg. Is that right? And where he, since 1994, since 1988, he's director of this institute. And actually, I got to know about his work early when I was still undergraduate student in Chile, and something that took my attention and to my eyes is a nice field where there is a binding, a join of a field of developmental biology and immunology, both conceptually. I was doing developmental biology, and I realized that it's different, the kind of question in comparison to immunologists, and here was a place where two things were joining. Also, his research, something that is important to say, is not really mainstream, so there are not many people who work. I mean, it's like his field is not like a hot field where many people are working, something that also makes it very special. And also the kind of questioning and the kind of research is like very, very long term. So it's not like, let's say, what I call a bit not nicely, like it's nothing related to McDonald's research that you can face today, right? And well, he will tell us about the evolution of the non-self discrimination in adaptive immunity. And of course, he has been studying, he has done very important contributions to understand the development and evolution of the immune system, especially the adaptive immune system, and focusing especially in the formation of the thymus, the organ, and how the selection and of potentially self-reactive cells exist. So with this, I want to leave the place to Thomas, and we look forward to your presentation.
Okay, thank you very much, Pedro, for this very kind introduction and the kind words. When he picked me up at the station, he said, "You're doing science in the old-fashioned way." It's okay, I'll explain. So I wasn't sure whether he invited me because or despite this, and I also hope that you won't be disappointed.
In terms of the big questions.
Okay, now I realize it's only half. Oh, well, never mind. So what I will be doing today is not to spend too much time on describing to you precise experiments and all the problems associated with them, but rather more give you some general idea of what the lab has been interested in the last 20 years perhaps, where we try to understand how self, non-self discrimination was deployed in the immune system during the course of evolution of that particular system. And what I would like to say is that self, non-self discrimination is of course not something that was invented for the immune system. The immune system simply used it for its purpose. Non-self discrimination, self, non-self discrimination is pervasive in biology. You can see that here. I took this from a recent paper in Science, where bacteria in one way or another can segregate based on some genetic component, so they can detect self and distinguish it from non-self, and then, as you see here in these pseudo colors, can nicely separate. Do we have a pointer? No. Okay. Well, the color is easy to see. You see mixing or unmixing. So if one considers this in terms of what the immune system really does, it, in the most general sense, it protects organismal integrity, and that is done by, in one way or another, discriminating self from non-self. Now, as I said, this is not only for the immune system. Another big area in biology or in biological systems that use self from non-self discrimination is, of course, in sexual selection. And I've summarized this here in this table, so you can see a recognition of self, or this divided being in recognition of self, recognition of non-self, and mechanisms that use self and non-self in this process. And what is important here is that I've found examples for all of these processes, both in pre-zygotic sexual selection, that is when you go and try to find a mate, and also in post-zygotic immune surveillance. So when the organism has formed, it then has to somehow decide whether it's self or non-self, what happens in tissues, and when microbes invade, and these kind of things. And for all of these different types, self, recognition of self, of non-self, and a system that recognizes both or deals with both, you find these pre-zygotic and post-zygotic mechanisms. So one can probably learn in mechanistic terms from this list, in the sense that the immune system most likely has not invented anything new, it simply adapted mechanisms that pre-existed for its purpose. So just to summarize this again, so the mechanisms in the immune system operate on these three levels. But the important point when one thinks about this is, of course, that there is, during the development, either in evolutionary time scales or at the level of the individual, when the individual evolves, any recognition system that you deploy to do this differentiation requires some sort of quality control. And I will go through a couple of examples just for you to get the idea why we are thinking along these terms. It's no rocket science, it's really trivial, but just to remind you. So if we consider recognition of non-self first, so the idea is for an immune system that some effector mechanism is activated when it recognizes non-self. So you might say, okay, that's a relatively easy way of doing it. If I have no constraints on coding capacity, I can develop receptors that in one way or another are not reactive to self structures. That can be done over Darwinian timescales. Then these receptors can recognize something that is not self to me. But of course, the coding problem is a big problem. You cannot develop receptors for non-self by unlimited genetic material, because that is a big constraint on this. This is the simple explanation why these germinal encoded receptors of non-self are, of course, pattern recognition receptors that sort of detect with one receptor many different types of microbes. So TLRs being perhaps a famous example of this. Now you can also turn the the coin and you say, okay, I'll operate by recognition of self. Now, of course, that poses a problem because the effector system has to be constrained. It cannot be activated by presence of self. It can only be activated by lack of self. So that, of course, again, poses the coding problem. But now you have to organize it in a different ways. Now you have to somehow instruct the effector mechanism that it only becomes fully operational once there is no self structure around. And how is that being done? You know the famous example of NK cells. They recognize, you can think of this in different versions, but NK cells recognize this via version 2, where they interact with polymorphic molecules. But they recognize the absence of self molecules, MHC for example, so they know there's something wrong with the cell, then they become activated. The important point being here is that Darwinian selection cannot work on these systems, because it has to be somehow somatically determined if you operate in this version 2 mode, which is a polymorphic molecule that you're recognizing. Because if it's MHC, it's likely to be different in all of us, so our general innate type of receptor system has to be trained, and it has to learn what it should see and what it should not see, and this is termed licensing, as you know. So it's a kind of quality control that the maturation of the effector cell is stopped if the receptor cannot see self, otherwise it becomes mature, and then it can, of course, act when there is lack of self. And you can also, of course, easily see that this is likely to be an inactivating type of signaling pathway. Now, to complete the picture of your little scheme on the back of an envelope, you can think of a system that operates both on the recognition of self and non-self. Here it, of course, is a little tricky again in terms of the coding system, and the solution that the immune system has found, or perhaps the immune system has co-opted from previous systems that were not connected directly to immunity, was the process of somatic diversification. Again, this is nothing new to you. We know that our T-cell receptors and B-cell receptors are somatically diversified within the lifetime of an individual in more or less random way by combinatorial and junctional diversity. And that is also true for the antigen receptors in the sister group of our type of vertebrates, the jawless vertebrates, where these antigen receptors are called variable lymphocyte receptors when they were discovered by Max Cooper's laboratory. But the general principle is that the outcome of this somatic diversification process generates some aspects of randomness that one has to deal with. It is perhaps not entirely random, but at least there is an element of indeterminate structure in this receptor repertoire, so one has to think about how one can control this. So somatic diversification means somatic quality control. Whereas in the previous examples, we could rely on Darwinian selection, of Darwinian quality control in the recognition of non-self, for example. Here it's mandatory that we do this on a somatic scale, on a somatic scale in the individual. So this is a very simple-minded scheme that you all know about. You have an initial repertoire of receptors. You probe them against self structures in various ways, sometimes very ingenious ways. But this quality control is linked to the site of production. It's not like the car manufacturers that sell you a car and later discover that the brakes don't work. Here it has to be coupled intimately to the site of production when the cells are released, because they are not reactive to self. They are either deleted or they are made anergic or in whatever way. But the outcome of this is a self-tolerant repertoire. Of course, there are little details to this, but that's sort of the overall idea. Now, if we have to couple production, that is somatic diversification, and then quality control, somehow physically, we have to generate this ideally in a confined anatomical compartment, and that is, for me, the raison d'etre for a primary lymphoid organ. So you you initiate somatic diversification, you coordinate it with quality control, and then in the periphery we only see what is a self-tolerant repertoire, made self-tolerant in many different ways, but functionally that is what we see. Now when one thinks about this analytically, it's clear that in this case the unit of selection is a cell. It's no longer an organism, a full organism like in Darwinian selection. It's a somatic cell, and the time scale is the individual. So we know that the development of this repertoire is complicated because we have this randomness in it. And now, of course, you could say, if all of my receptors that I'm making are self-reactive, and I purge them from the repertoire, the final repertoire of self-tolerant receptors is nil. That, of course, is not the way of doing it. So one has to think of ways how one can balance the degree of potential self-reactivity against the size of the peripheral repertoire, because the peripheral repertoire, or the diversity of it, rather determines the immunocompetence. That is the way the organism can respond to a challenge from the outside. Now it turns out, and I'll give you, without going into too much detail, it turns out that you can calculate this again on and back of an envelope, that monoallelic expression of antigen receptors is the solution to this problem. There you can have the optimal solution. So this is my summary here, it's in small print. It's monoallelic expression of these potentially self-reactive receptors that ensures both the greatest diversity and the greatest number of cells that survive this quality control. Now a bit of would have needed a pointer, but you can see here, this is the fraction of self-tolerant effector cells resulting on the y-axis, and as a function of the probability of self-reactivity. So if self-reactivity is 1, that means all receptors are self-reactive, the repertoire, of course, is 0. And you can go, you see this is a logarithmic scale, you can go, and that depends on how many receptors you express on the cell surface. Clearly, if you express many receptors on the cell surface at a certain probability, the chances that the cell will be eliminated by this process is greater than if you only express one receptor. And this is now meant to be one gene that is active, and you can continue this calculation. Here you see the number of different receptors in the final repertoire at the bottom, and you can extend this even a little bit further when you go from biallelic to monoallelic expression. This is the optimal solution for a system that generates receptors with elements of randomness, if we think of the final diverse repertoire. Now, of course, there is many problems that one can study. There are very small vertebrates that don't have enough lymphocytes to even think of representing the entire, at least potential repertoire. There are very big animals, like the blue whale, for example, and one would like to study, of course, the immune diversity in these animals, but I think it's never been attempted. It will actually be technically quite difficult, but scientifically very interesting, I think. Now, if we think of this process and the organization of the quality control, this quality control requires some cell non-autonomous interactions, because the receptor is expressed at the surface, and now this cell has to be interrogated. So this non-autonomous interaction takes place in the primary lymphoid organ. And when we think about this, this is perhaps the motivation why we studied, like Pedro said, why we study the thymus, which is the primary organ where T cells develop. This has two aspects to it. One is a morphological basis, that's the organ as such. And the thymus is good because it's anatomically very discrete. Even for a medic like me, it's easily recognizable. And one can take it out and one can manipulate it. And there's also a mechanistic basis for this process. So we need to find ways of probing the specificity of the receptor. And ideally, this is the same system that is being used in the periphery then to initiate an immune response. And it would be interesting, and this is what I will be talking about in the next few minutes, it would be interesting to consider the evolution of these two aspects and see how they somehow are intertwined during the 500 million years of evolution of vertebrates, for which we know now in these two sister branches that they have a similar type of immune system, meaning that they generate lymphocytes as effector cells of different types, T and B-like cells. They use somatic diversification to diversify antigen receptors, albeit they're of different molecular structure, but the general principle, the design of the system is the same. Now let's consider the morphological basis first. So as I said, we are interested in the thymus, so we work on the thymus, and we are particularly interested in this context, and for various other reasons, in the microenvironment that fosters the development of the hematopoietic cell. So the hematopoietic cell is our effector compartment, and this non-cell autonomous control part, the quality control, happens via interaction with the microenvironment. Now just to give you an idea what thymopoiesis means and how this process in itself during development somehow depends on provision of both parts in the equation, I show you a video that Isabel Hess, a postdoc in the lab, made some years ago, which utilizes a zebrafish which can be put under the microscope and watched for extended periods of time. And because T-cell development and thymus development, in aggregate called thymopoiesis, in the zebrafish begins relatively early during development, the fish are really very small. You can put them in agar under the microscope. They grow while you're observing them, and you can observe them for 24 hours or even longer. Now, what we've realized from this is two things. First of all, there is some sort of autonomous development of the microenvironment, and that is here indicated in these still photos. You see, I didn't explain this, actually I have to go back. So we are marking now the fish in 3 colors. The microenvironment is in red, that's the thymic epithelium. We are marking the endothelium of the vessels in blue, so we get an idea of the vasculature in the embryo. And we mark the hematopoietic cells, or the ones that are migrating to the thymus, mostly lymphoid progenitors, and some other cells too, with GFP. Now, when you look at this still photograph, you can see that in the 3rd day of development, there is a small patch of red cells indicating that the microenvironment develops first. You see that a little later, this is in this still a good hour later, there is the first lymphocyte approaching that particular microenvironment, guided by chemotactic cues. We don't go into the details, but you see it's a directed migration. And the next time point, you see how these cells then make contact. This is a nice representation of what has been termed a lymphoepithelial interaction. This is how the cells make contact. They receive specifying signals to turn into T cells, and later on they receive these quality control checks whereby MHC molecules expressed on these epithelial cells probe the T cell receptor antigen complexes and see what they can make out of it. And the next slide then of course means that all the friends are coming and they are coalescing, and while they are coming, the microenvironment develops and differentiates, and it will not differentiate fully without the presence of these hematopoietic cells, indicating that there's mutual interdependence. Now, I run this video now, you will recognize these features, I hope at least. So you see the vasculature very easily, and now I cannot point. But you will see our pioneer lymphocyte coming, and that shows you the direction of where you can see a patch of red cells emerging in the middle of the picture. And there is a pioneer. Look, divides, and it approaches this red patch of cells. You can see that now. So this is a very dynamic process, and because in this particular organism one can interrogate this process by genetic interference, one can get a good idea of which kind of genes and molecules then are actually involved in this particular process. So the bottom line of this video and all the investigations that surround this kind of work is that the key thing in this process is that the developing T cells make contacts to the microenvironment. That is the quality control check point in the car factory. Now, we know now We know now that somatic diversification of antigen receptors happens in the two branches of vertebrates. That is in the left branch, that is the agnaisons, the jawless vertebrates, where there are only a couple of hundred at most species. And in the other branch to which we belong, there are about 70 or 80,000 different species. So it's a bit disproportionate, but nonetheless these two branches have survived, and they presumably originated from some common vertebrate ancestor that's depicted in this little round. Huh?
And also use the arrow of the...
Yeah, I'm not very good. No, I'm not very good at this. Then I always click. So that forces you to look actually at the pictures that I'm showing. Okay, so this little round in the middle where these two branches come together, this is our fictitious, now no longer living common ancestor of all vertebrates. And below that, in our evolutionary tree, are cephalochordates. I'm just mentioning this. This is the basal type of chordate from which we all spring during 500 or so billion years of evolution. Now, if lymphocytes are present in all vertebrates, we should assume that the quality control system that we think we know about from studies in mammals should exist in all vertebrates. So we can simply make that assumption. And we can also assume that there is a developmental program that specifies this particular type of tissue that is capable of affecting the development of these cells or directing the development of these effector cells, lymphocytes, and at the same time coordinating quality control. One can even think in various steps, and I'll come back to this, that this process evolved in steps, that perhaps the primary or the evolutionarily ancient version of a primary lymphoid organ was perhaps simply a microenvironment that fostered the development of lymphocytes without worrying about somatic diversification of receptors, just a specialized area in the organism where, in our case, lymphocytes would develop. So lymphocytes simply liked it there, they developed. And then, as time went on, and perhaps somatic diversification was invented, again, I will conclude with this, then there was an extra need from within the microenvironment to ensure that quality control would be effected in the same place. So why not look at transcription factors that control the development of this very special microenvironment, this very special type of epithelial cell? And many years ago, almost, actually more than 20 years ago, we discovered a transcription factor by looking at a natural mutant that occurred in mice and in rats and in humans that would lead to failure of T-cell development in the thymus. And this transcription factor goes by the name now of FOXN1. This FOXN1 is depicted here as being present in all gnathostomes. That is the right-hand path of our developmental tree of vertebrates. It's also present, just a nomenclature issue, in jawless vertebrates. This is the next line down. And then, interestingly, there is another gene which for nomenclature reasons has been given the name FOXN4, which is the evolutionary forerunner of FOXN4L and FOXN1 in this particular picture. So these two thymopoietic master regulators have arisen through gene duplication from an ancient gene that you can see goes down way down in metazone evolution. Now, having identified the function of this so-called master regulator in mammals, we wondered whether this is actually also doing its job in earlier, in more ancient forms of vertebrates. And that is indeed the case. I'm not showing you these results. But we can go one step further. Now we go into what probably Pedro meant that we are working outside of the mainstream. Now we go into fantasy. And the fantasy is that we simply hypothesize that the evolutionary history of these factors somehow must have predated or antedated or anticipated future function in the way of preparing a particular type of tissue. So I'm just giving you an example here. So this FOXN1 gene is already very nicely expressed in the in the thymus of a cartilaginous shark, which is at the bottom of our Natterstone lineage. It's also expressed in the thymus equivalent of jaundiced vertebrates, of lamprey, for example. I'm not showing you that example. But the key point I would like to make now is, if we now go away from vertebrate and we go back in time another 50 million years perhaps, and we now focus on basal chordates. These basal chordates have, as you can see in this line here, they have the ancestor, the precursor of our thymopoietic factors. Now, because it's a transcription factor, and transcription factors tend to evolve in various ways, and we have detailed information about how this actually happened, we can easily study their function by removing them from their original context and placing them in a, let's call them, modern context. And I will show you this experiment because I find it very instructive. The caveat, of course, is that we are doing something that is not natural. We are just accelerating now the function or the duties of a transcription factor that evolved 550 million years ago and now bring it to modern times. Maybe this is asking a bit much, but we were encouraged by finding that this Amphioxus version, which is actually this cephalochordate version of the ancestor of our thymopoietic factor, is already expressed in the pharyngeal endoderm, as you could glean from the right-hand section in a early cephalochordate larva. So it already is expressed in the right place. And then, of course, we consider the possibility that perhaps it is there and marks something. Of course, it does not have the role of coordinating lymphocyte development because these cephalochordates don't have lymphocytes, but maybe they have sort of similar type cells, we don't know yet. But they certainly don't have a quality control because there are no known rearranging or somatically diversifying antigen receptors. But something is there. So already the expression domain of this gene is fixed in chordates. And that, for me, is of course an argument to say this is why the thymus is where the thymus is, because the ancestral gene just happened to be expressed in the region where the other genes maintain the expression domains, and this is why the thymus is there. The thymus functions everywhere because it's not autonomously seeded by migrating hematopoietic cells. You can put it under the kidney capsule, as you know, or anywhere else, and it will always be functional. This is just an evolutionary coincidence. Now the experiment is a very simple one. We take a mouse, we remove the thymopoietic factor, that is FOXN1, we create a knockout, and we replace this endogenous modern, let's call it modern gene, by this ancient cousin. And then we ask what happens. Now I remind you, when you see the result in a moment, I remind you that our fictitious sequence of events is that lymphocytes developed first, then came somatic diversification, and alongside somatic diversification, but perhaps a little before that, was a preparatory phase that would allow a very quick installation of a quality control. That poses another problem that I will address in the next part of the talk. But now to this experiment. So this is the scheme. We take out FOXN1, this dotted line. There's no thymus development, and we replace it with the endogenous one. That's fine, of course. And now comes the test, which is this cephalochordate cousin. So what does it do? Surprisingly, it works. You can see the normal replacement of the wild-type situation on the left. You see CD4 and CD8 cells for the aficionados. That's and the big blob in the middle is double positives, immature thymocytes. In the non-functional rudiment, that is in the epithelium that cannot support T cell development, of course, there is nothing. And then on the right-hand side, you see the outcome with this ancient cousin. It makes double positive cells. It supports the development of cells to the stage where they express their antigen receptor, and then it fails. No further selection into single positive cells. And that reminded us of an experiment, another crazy one, which was that we thought, by looking at the gene expression profiles of these various components, that we could reconstruct or reconstitute the processes that make a thymic epithelium, a thymopatic environment, a thymopatic environment. So which genes are required? So we started this experiment by looking at a FOXN1 deficient rudiment, and instead of replacing now the transcription factor as a whole, which then of course coordinates the whole sequence of events, we took presumptive target genes and replaced them one by one. And I just show you two combinations. One is a combination of two chemokines, CCL25 on the right-hand side, a notch ligand, and stem cell factor, and that produces very nicely the outcome we would expect for a late embryonic thymus. It even works with only two factors, that is the CXCL12 and delta-like 4. And that means you don't only need a chemokine to attract these progenitors to your site of action, as it were, a T-cell specifier. And these cells that you find in this upper quadrant in the left corner, they express a diverse repertoire of T-cell receptors. So they actually make the T-cell receptor. But then they stop, because in this particular reconstitution experiment, there is no MHC that is important for selection. So these cells, once they are produced, then they die, because they need the interaction, and the T-cell receptor needs to signal into the cells for them to survive. So because they have nothing to see, the T-cell receptor has nothing to see, the cells die. But it shows in principle that one can reconstruct this particular thymopatic environment, and we know that this transcription factor that I just showed you, this very ancient one that didn't know that in the future its descendants would be able to organize T-cell development, can already do these early steps, but not any further. So this is our, again, fictitious scheme, so that we have two parts to it. We have a lineage determination and a stage where the cells make a receptor. But then we need the interaction of cells to coordinate quality control. And in the advance from a non-vertebrate, the addition was a chemokine, because these cephalochordates don't have any chemokines. So this system would not have worked to bring cells to a particular place. And in the early vertebrate, that would then work. We bring in the chemokines, and the first stage is met. We would produce cells that produce a receptor, but there is no further selection, because that is not required, we assume, because there is no somatic diversification. Now I can put this in a big scheme, and you say, okay, what are the innovations? The innovations that are apparently, or have apparently taken place in the transition to vertebrates, was somatic diversification. For whatever reason, all of a sudden, all vertebrates wanted somatically diversified antigen receptors. That was the innovation, and the microenvironment must have acquired, in addition to producing cells with a receptor, now add more facilities to allow for quality control. That brings us to the mechanistic basis of it. Of course, you all know this. I tried to put it into a slightly different perspective. So we know that the T-cell receptor recognizes antigen in the context of MHC. So MHC-mediated peptide presentation is at the heart of all of this. And these self-antigens that are being generated in the thymus and presented to the developing T-cells, of course, then purge the repertoire of self-reactivity in one way or another. And then when these peptides bind to MHC in the periphery and they are not recognized as self, they initiate an immune response. Now, in abstract terms, this is an intercellular discrimination, because one cell presents the antigen, the other cell recognizes it. It's like a sensory modality. In this case, the T-cell receptor senses and recognizes this particular complex. And the components that are being seen are intracellular. So in the cell, as you know, there's peptide, protein degradation, and